Hello everyone. Mrinal and we were working on a project and the name of our project is Identification of Pathway Leading to Ototoxicity by the Use of Cisplatin. So before diving straight into the topic, I would like to tell you briefly about cisplatin and ototoxicity. Cisplatin, it is a well-known chemotherapeutic drug that has been used for the treatment of numerous human cancers including bladder, head and neck, lung, ovarian and testicular cancers. Cisplatin exerts anti-cancer activity via multiple mechanisms, but its most acceptable mechanism involves the generation of DNA lesions by interacting with purine bases on DNA, followed by activation of several signal transduction pathways, which finally leads to apoptosis. Cisplatin induces hearing loss, that is its side effect, and it is usually bilateral and irreversible and is particularly serious in pediatric population with cancers like neuroblastomas, CNS, head and neck cancers, while irradiation of the base of the skull of brain is also performed. Now, I would like to tell you about ototoxicity. What is ototoxicity? It is basically when a person develops hearing or balance problems due to a medicines. So here our medicine is cisplatin, and it can happen when someone is on high dose of a drug that treats cancers, infections, or other illness. So, so the specific aim of our project is to identify the genes and pathways related to the side effects of cisplatin using the gene expression data from cisplatin treated lung cancer cells. Now, for that, we took a data set for which the geo accession number was GSE14814 and its title was Prognostic and Predictive Gene Signature for Adjuvant Chemotherapy in Resected Non-Small Cell Lung Cancers. For this, we took 133 samples, out of which ADC were 71, SQCC were 52, LCUC were 10. ADC stands for adenocarcinoma, SQCC stands for squamous carcinoma cells, LCUC stands for large cell undifferentiated carcinoma. So now I would like to hand over it to Mrinal and he will tell you further about the project.
Thank you, Yachita. So further from the data set, we did the principal component analysis or the PCA on two conditions. Like one was the one with all over samples, including ADC, LCUC, and SQCC, and other one was with the ADC and SQCC only. And while doing the analysis, we found out that the data distribution of the ADC and SQCC was made much better than the original data. So we focused our analysis on ADC and SQCC samples only. After finalizing the group of ADC and SQCC, we did its differential expression analysis on MS Excel, where we calculated the p-value using the t-test. From the t-test, we calculated the false discovery rate or FDR, which was then sorted on the basis of less than 0.05. And after that, we calculated the log fold change value, which was filtered on the basis of greater than 1 and less than -1. When all these parameters were met, we found out that out of 13,514 genes, we got only 7,386 genes. For the functional annotation, our 7,386 probes were then were significantly differentially expressed between ADC and SQCC samples, which were then functionally annotated using David annotation tool, and enriched pathway were identified by the human Gage pipeline on the T2 bioinfo server. As we can see, these are the results from our human Gage pipeline, and most of the enriched pathways are tumor-related pathways with significant upregulation contributing genes. The whole enrichment shows that even for pathways involved in tumorigenesis, different tissue types will always have different expression patterns. And significantly, our glutathione pathway is a common defense mechanism against the cisplatin-induced upregulation of the ROS or NOX3 gene, which are highly implicated in the ototoxicity. And for the David functional annotation result, after our human Gage pipeline, the 7386 genes were then uploaded on the David annotation, from where we found out 215 pathways, and out of those 215 pathways, we selected top 20, out of which we found out that only 13 caused ototoxicity, and whereas the remaining 7 does not cause the ototoxicity. From all these, we had the conclusions like several genes were identified which were differentially expressed between ADC and SQCC lung cancers with their morphological differences. Most of the genes and associated pathways are related to the cell differentiation, tumorigenesis, and DNA replication. Among the enriched pathways, we also discovered that glutathione, a metabolism pathway that is enriched, this pathway is implicated in a form of a defense against the cisplatin-induced ototoxicity in cells from the human inner ear. We also showed that it is possible to identify specific gene expression patterns and pathways related to side effects of cisplatin treatment in the lung cancer. Thank you.
Thank you, everyone.